You finally sit down with your book, but then you realize you forgot to take out the trash. And that's when you wish you had a clone, which in science fiction is an exact copy. But in biology, this isn't the case. Let's start with the basics, or DNA, or deoxyribose nucleic acid. DNA has all the information our body needs to create proteins that help us maintain homeostasis, give us our traits, and basically allow us to function. Clones, in simple terms, are organisms with the exact same genetic makeup as the original organism. Twins are a good example of natural cloning. Here's how artificial cloning works. Say this is person A, the person whose genes we want to clone. We extract somatic or body cells from person A. Somatic cells are cells that have copies of chromosomes from both person A's mother and father. From person B, we extract an egg cell, which is a gamete. To proceed, we take the nucleus out of the somatic cell. The nucleus is the segment of a cell that contains the genetic material we want the clone to have. Then from the egg, we extract the nucleus. So now the egg becomes a vehicle for the genetic material we want our clone to have. Since this process is artificial, we need to apply an electric shock to the cell to trigger it to divide. The resulting cell undergoes cellular division and develops into an embryo. This is called somatic cell nuclear transfer. As you can see, this process doesn't require any external fertilization from a sperm because the nucleus that we're using from the somatic cell already contains the correct number of chromosomes. Now, these cells can become an embryo. The embryo that we now have from somatic cell nuclear transfer is put into the uterus of another female. When the female gives birth, the baby that is born has identical genes to person A. Now, of course, the clone won't be exactly like person A. Someone's memories, experiences, and lifestyle choices make up the way a person acts. They may look the same, but they don't have the same emotions or personalities. Human cloning is illegal in many countries because of the ethical concerns. One is that when we take out the nucleus of the egg cell, we are pulling out important proteins for cell development, so many embryos die before implantation. Another issue is premature aging and death. Telomeres are protective ends of our chromosomes that protect our genes, and they shorten every time the cell goes through replication, eventually dying when they are too short. With this cloned organism, the telomeres start off short, so the organism is at risk for premature death.